Hi, everybody. I'm Michelle Tennant Nicholson, the president of Henderson County Beekeepers Association in beautiful Western North Carolina. You may know us as the Smoky Mountains. I was recently in Pinehurst, North Carolina at our state conference with the North Carolina State Beekeepers Association, and I was so thrilled to see all these women on the stage with Dr. Tarpey. Now, for those of you who are like, who's Dr. Tarpey? Here in North Carolina, many beekeepers are raising funds to do for an endowment under Dr. Tarpey for beekeeping research. So when all of these young women who are doing research that's related to beekeeping, it's just so very exciting because so many of us have been doing fundraising and really committed to this endowment. After the conference, I thought, what a great topic for our YouTube channel. That's where we have Eva. Now, so how do you say your last name? Is it Corral? Help me say your
Corral.
Corral.
Corral. And first name is pronounced Eva, actually.
Eva Corral. Did I say it right?
Yes.
Perfect. So Eva is going to be talking to us about heat, virus, and drone viability. And I'm going to let her introduce herself so that I don't butcher her credentials like I did her name. Eva?
Hi, yeah. So my name's Eva. I'm an undergrad student at the University of Colorado in Boulder. And this summer I'm doing research at NC State in David Tarpey's lab. I'm working with Bradley Metz, who's also in that lab. And we're researching drone sperm viability in the face of a bunch of different, or just two different factors, which is heat and virus. Um,
Oh,
so
well, I'm
yeah.
excited about this. I'm excited about this. And it's a little bit of a sexy topic because we're talking about sperm. So if that offends you, just move along. But let's get into it.
I always have to put that disclaimer before I start. Just letting you know, I'm going to say sperm a lot. But
Awesome.
yeah, so let's get into it. Let's see. I don't know how to go to the next slide.
There
There
you
we go.
go. Here you go.
Awesome. Yeah, so in terms of virus and heat, the virus that we worked with this summer is deformed wing virus, which I'm sure all beekeepers are aware of.
We are.
It's a pretty brutal one. It's just everywhere. So yeah, that's why we chose it because it's so common. It affects almost every single colony, if not all colonies, that anybody keeps. And we combined that with heat. And so this little graph that I drew out is actually, it's just kind of showing what happens once heat is added to the sperm outside of the drone. So we have a drone with or without virus added, and then the viability, no matter what, is going to start at like a certain point and then drop down over time in the face of heat. So our time course is like 4 hours, and once you get to this end of the 4 hours, it's going to be low. But the question is whether or not the viability is going to drop at a faster rate or a like less steep rate. Kind of supporting two different hypotheses that people have had about it, like either there's this immune system response called heat shock proteins that might be made in the face of virus or heat stress or just different stressors. And so there are two theories of either those are made and it makes the sperm a little bit more robust, or they're not made and the sperm viability just tanks. Uh,
Okay.
yeah. So then to do all that, we first went out to our hives that we have at NC State, collected 90 drones for our sample size, and then we injected a few of them with nothing, just like kind of a placebo sham injection. 30 were just like a control, so they were not touched at all, and then 30 were injected with our virus. So they're all mature drones. They're sexually viable at this point. We try not to take any young ones out of the nest. And then we incubate those for 3 days, let the virus kind of run its course. And then after that, we dissect them and we analyze their sperm by taking out their seminal vesicles and adding that to a solution that kind of highlights the live and dead cells. And it's a really cool thing. It like you put it into this machine called a salometer, and it will count like live over dead count and tell you like the sperm viability. It's like a percentage. So yeah, we do
And
that. And
did you draw these cartoons?
I did, yes.
They're very good.
Thank you. Yeah, I
Yeah.
couldn't find the visuals I wanted, so I was like, strong.
That's perfect. Okay, yeah,
Yeah,
it's really clear. It's very clear what we're doing right here. And so,
good,
I have no questions yet, but I will.
good. Okay. So yeah, then once we did that, we analyzed all of these sperm viabilities at different time points, and this is essentially our results graph here. So you can see they all start at a similar viability. There's like a little bit of variation in our control group, but generally we've got this even viability. And as you go down towards the end of the 4 hours, that it obviously declines. But what's really interesting is our virus infected group actually stays higher on that 2 hours. That's our like most significant different point there on the graph. And this is the average of two, really like our two reps of the experiment that gave us like very clear results.
Hmm.
So yeah, our sham and our control are kind of down here, similar-ish. The sham we had a bunch of issues with, but it stayed pretty similar to the control. And then our virus, for whatever reason, is higher. So that kind of supports our more resistant theory that I talked about earlier. So
Hmm.
yeah, those are our main results from the study. We did a few reps over the summer in the past few months, and the only reason we had to stop was the drone season kind of is coming to an end. It was so hard to find these drones. We were out on like 100-degree weather, just like hunched over picking drones out of it was brutal. So yeah.
So and how will this apply to, a backyard beekeeper's drones? Like how will this research impact the everyday beekeeper?
Well, it's kind of what I'm doing is part of a larger study that Bradley Metz is working on within David's lab. And it's kind of just analyzing like how is reproductive viability like changed by a bunch of different factors. So he wants to talk about or look into like age, drone size, and then reproductive viability like in the face of virus and just a bunch of different stress factors and see like really how drones are responding to a bunch of new issues like climate change and these viruses that are being introduced. So it's not super applicable in like every day. It's definitely a lot more in like a reproductive science thing, but I thought it'd be interesting to just think about like when you see the drones, like they are doing like they're so clearly made for one thing, and that is reproduction. So like even when they're infected with a virus like deformed wing virus, like their body is making sure to double down and protect that. Yeah.
It's really fascinating. So the other, the macro conversation here on a global scale is when we think about different pests like varroa mite,
Mhm.
one of the theories that we can actually fight against varroa mite would be genetics and actually having genetic choice in our queen rearing. And so this one of, and of course, we always welcome and love the research that's coming out of Dr. Tarpey's studies and cohorts. But the one thing for me that's really exciting is being able to control in for a backyard beekeeper or commercial beekeeper, how are we actually grafting queens, selecting for those qualities for the type of bee that will then be able to fight these pests and these viruses. And more importantly, information about heat is important, right? There was something, Eva, that I heard a speaker many years ago say, and we were I remember hearing an audible gasp in the room of beekeepers because she was talking about, did you know that if you expose your brood, even while you're doing you're looking at your frames, but then you've exposed your brood, you're sitting there looking at it, and let's say it's a certain temperature outside. She had done research on how that little bit of exposure while we're doing our inspections can actually stunt the growth of that brood. And
Oh
so
wow.
she was specific on the temperature.
Mhm.
So when we look at heat or cold, you remember that our bees are really keeping a sustained temperature inside the hive. And
Yeah.
so this type of research helps us as beekeepers understand how hot and cold impacts our choices on having the strongest hives possible. So
Mhm.
that's one reason why when I was in the audience and I saw you present, in Pinehurst. I really thought, gosh, this would be really very fascinating for beekeepers at large. So what's next for you? Are you gonna be a beekeeper or continue on with your research, or let us know what can we expect from you?
Yeah, I mean, I before the summer haven't been like I don't keep bees on my own. I have relatives that definitely do. And so that's kind of what got me interested in this internship in the beginning. But I'm surrounded by a lot of entomology girls. And I originally am not, but I'm gonna be a junior in college next year. So I have time to kind of decide if I want to go the entomology route because the summer's been really, really just like super enriching and like awesome for me. So yeah, Sammy Ramsey actually is at CU Boulder now.
Woo,
And
girl, come on.
yeah, so I'm gonna maybe go and see if I can chat with him, get some good information from him,
Uh,
or even get involved in his lab.
For those of you who don't know who Dr. Sammy is, Dr. Sammy is the person who's really credited toward discovering that Varroa mites feed on the fat bodies of the honeybee. So he's sort of a big deal for us. So if you're like, ooh, why is that a big deal? Well, it's important because in our fight to have healthy hives and help our bees, researchers like Dr. Sammy make a huge difference in our ability to have medicines and information, everything that we've really, the reason why we're forming clubs and associations so that we can share this type of information, the reason why we exist. So that's really exciting. And just
Yeah,
love Colorado. If I am not here in Asheville, North Carolina, I love to be in Colorado. So it's just, another state that's, I hope that you do take on beekeeping because it's really
yeah,
great. And then now that you know about heat, you can keep your bees overwintered a little more,
definitely.
a little more consciously, right? Because that's
Yeah,
going to be tough during the winters for those beekeepers for sure. Well,
definitely.
great. Well, stay in touch with us. And how can people be in communication with you? Like, how could they? What socials? Like, where can they connect with you?
I am on LinkedIn.
Great.
And it should just be Eva Corral spelled how it is on my um thing here.
Perfect. I'll put them also in the description, everybody. So I'll look up her LinkedIn profile and then
Yeah.
link it that way. And of course, we're at hcbeekeepers.org, Henderson County Beekeepers.org, and our state association is ncbeekeepers.org, North Carolina Beekeepers.org. Thanks so much for being with us today, Evan.
Thank you for having me.
Good luck to you.
Thanks.